front row to sit down. Oh, everyone, please stand up. Okay, I'd like everyone in the front row, including Claire and Charles, to sit down. Everyone in the middle row, sit down. And you two, sit down. So let's count Leslie as standing right now. You guys represent the population of people, of Americans, who have metastatic breast cancer. If our class had, as a whole, had breast cancer, and our school had cancer in general, well, our grade. Now, I would like you guys to sit down. Leslie, you're sitting down too. Okay, Mr. D represents the population of Americans who has metastatic HER2 positive breast cancer. And so Charles would walk you through the numbers, actual numbers. Mr. D can sit down.
So 1.6 million Americans have breast cancer in 2000, or were diagnosed with cancer in 2012. Out of those 1.6 million, 229,000 of them had breast cancer. 30% of those people who had breast cancer had or metastatic breast cancer, which means that their cancer had spread to other parts of their body, which is just stage 4 cancer. And then of those people, 25% of them had HER2 positive cancer, which means they had overexpression of the HER2 receptor, which is a growth receptor, and basically they had 100 times more of that receptor than a normal cell would. So in total, out of all Americans, about 17,175 people had cancer, had this type of cancer that are drug targets, which is HER2 positive metastatic breast cancer, in 2012, which doesn't seem like much, but it's still a good amount of people.
Okay, so the drug we chose to research is Kadcyla, which research was done by Genentech. And Kadcyla is an antibody drug conjugate. So has anyone ever heard of the term antibody before? Just raise your hands. Okay. Can anyone give me a definition? Okay, Mr. D.
Do you want me to?
No, it's okay. So an antibody is just going to be a specific protein that targets an antigen, which in this case is going to be the HER2 receptor found in the cancerous and healthy cells. So that part is just like the purple part here, which is the trastuzumab. And then the other part of the drug is a chemotherapy drug, which is the DM1 or mertensine. They're going to be the same thing, but we're going to call it DM1 for the rest of the presentation. Okay? And then finally, it's just like the link that holds the two parts of the drug together, which is the MCC here. So you just have an antibody, which is the trastuzumab, and a chemotherapy drug, which is the DM1. So what makes Kadcyla really interesting is it combines those two things together. And Kadcyla was FDA approved on February 22nd, 2013, and it's given by IV infusion every 3 weeks for about 30 minutes. The phase 3 trial that got Kadcyla approved, FDA approved, was the Camellia trial. It had about 991 total participants, and they were either given Kadcyla or they were given a combination of 2 previously FDA-approved drugs, and that was lactinib and capecitabine. All of these patients had HER2 positive metastatic breast cancer. So all 991 people had HER2 positive, which means they're overexpressing the HER2 receptor, and their breast cancer has spread to other parts of their body. So what you're looking at in this graph is just how long they lived while taking the drug. So this is percent of people that are still alive. So at the 50% mark is what you're looking at. So gray line is the other treatments, and Kadcyla is the purple line. So you're looking at about a 5-month longer life expectancy while taking Kadcyla. Does that make sense? Okay, so there are also some side effects that came along with this, including fever, headache, nausea, and other stuff that I won't list all of them. And this is because Kadcyla also targets healthy cells, but we'll get into that next part.
Okay, so Kadcyla targets HER2 specifically, is the receptor that targets. Now HER2 is part of a family of receptors. There are 4 of them. There are human epidermal growth factor receptors, which just means that they promote growth in the cells, cell proliferation. Now, HER2 is activated usually by dimerization, which is just it combining with another protein that is similar to it. But instead, Kadcyla, or specifically the trastuzumab part of Kadcyla, which is the bigger part, binds to HER2, and it does 2 things. It flags the cell for destruction by immune cells, and then it also inhibits pathways such as the MAPK pathway, which activates cell proliferation. Yes.
So the trastuzumab is what prevents dimerization?
Yeah, that's the big one.
Right.
So MAPK pathway normally activates cell proliferation. So when Kadcyla binds to it, it inhibits cell proliferation. But first we need to know that Kadcyla does bind to HER2 specifically. So 4D5, just for reference, is the mouse version of trastuzumab. It's like what it was in mice. So when they did tests on mice, that's what it was, and then they transferred it to humans, and it was called something else. So here they measured like pairs. They put 45 in the cells, 108, and then this. I don't know what it is, but and they measured the pairs that bound together, and HER2 and 45. It shows that this binds 45 or trastuzumab binds specifically to HER2. Anybody have any questions on that? No.
What does the darker dot mean? Is that
It just means that there are more pairs.
more pairs?
So it shows that 45 did bind to HER2, not something else.
The Kadcyla is supposed to be binding with the HER2.
Yeah, Kadcyla is supposed to be binding to HER2, and the first part of Kadcyla is trastuzumab, and 45 is trastuzumab. Any other questions on that? Okay, so a bunch of cells here were given, well, they all started at 0 cells in a bunch of different plates. Each one of these dots is a plate. And they were given different amounts of 45, and then this to compare. And 45, the less cells were made, which shows that 45 or trastuzumab inhibited cell proliferation. So less cells were made if there was trastuzumab in the cells. Yes, sir.
Can you go over how the drug binds to the receptor and like what it does exactly?
Okay, so trastuzumab, which is the antibody part of
it's
Kadcyla.
one third of the drug, right?
Yeah, more than that. It binds. It's the biggest part of it. It binds to HER2. We actually noticed a problem with that, and that how does it bind just HER2 positive cells instead of normal cells, because they both have HER2 receptors, just HER2 positive has overexpression. And we called the company, they had no idea, so it must bind to both, which is what Claire was talking about before. It's a problem with their drug.
And then it turns off the MAPK.
It turns off, it inhibits the MAPK pathway, which is cell proliferation. Yes.
What is 45?
45 is just the mouse version of trastuzumab. Any other questions on that?
So it's the mouse version of
Trastuzumab, which is one of the parts of Kadcyla. Any other questions on this? No? Okay.
So, how does DM1 fight breast cancer? Put simply, it inhibits tubulin. Well, what does that mean? Trastuzumab with DM1 attached will first bind HER2 receptors on breast cancer cells, and it's taken into the cell through receptor-mediated endocytosis. The cell breaks down this combination and releases DM1 into the cell, and it attaches to microtubules. The DM1 attaches to microtubules, blocking the polymerization of tubulin. Tubulin is like the building blocks of microtubules. Tubulin can't be added on to it anymore, the microtubules. So they can't add on. So if microtubules play such an important role in the cell, can anyone tell me what happens if they can't continue to grow? John?
Cancer doesn't spread.
Exactly. The cells are stopped in their tracks in their cell cycle, and this cancer just stopped. So does anyone have any questions?
Are there any side effects that come along with that?
Well, Claire already
Yeah.
walked through that.
Oh, well, that's a result of the
Yeah, it's a result of like this and targeting healthy cells as well as cancerous cells.
Okay, so DM1 is part of the drug.
Yes.
Okay, so how does it inhibit the microtubules? Like how is tubulin related to the microtubules?
So tubulin is a monomer. And you guys know what monomers are, right? And microtubules is a polymer version of that. So DM1 acts as a competitive inhibitor. Like think of a train, and then this is a wall. It blocks you from adding any more cars.
So would the DM1 also inhibit the proliferation of healthy cells too?
It should, yes.
Any other protein that checks and regulates the growth of microtubules that may interfere with the drug?
We don't know.
What was the question for? What was the question?
Is there any other protein that may because there are different proteins that check the structure of something and tries to fix them or things like that. Yeah, well that stops something. So I'm saying, is there a protein that fixes the microtubules after
We didn't see anything in the papers we looked at that takes away DM1 or anything about it.
Okay. So that's the end of our presentation. This is like a summary of everything, so we're just gonna leave this up while we answer questions. That's it.
Clark.
so your drug inhibits MAPK pathway, right?
Yeah.
So there are a lot of other pathways that lead to cell proliferation of normal cells and cancer cells. What does it do with those pathways, or are they still active?
It inhibits the PI3K pathway, which is for cell survival also, but this is the main one that we encountered that drug
And
inhibits.
because we're looking at HER2 positive breast cancer cells, that's the pathway that's causing the problem. Because they're
There's only
overexpressing
one pathway causing the problem.
because they're overexpressing the HER2 cells, and that's the MAPK.
Okay.
Yeah.
So does Cancilla, does it limit cancer growth, or does it try to?
It mostly inhibits cancer cell growth and proliferation, but then the papers we looked at said that it flags cells for immune cells, for destruction by immune cells, and we asked the company that too. They didn't really know. They just said that happens. We didn't see proof of that, but so we did see proof that it inhibits cell proliferation.
So it's a dimerization of HER2 that leads to the pathway.
Yeah, normally HER2 dimerizes with another HER receptor, so HER1, HER3, or HER4, and that activates the pathway normally. But when Kadcyla binds to it, it inhibits that pathway. Being activated. That makes sense? Yeah.
DM1 is a competitive inhibitor to microtubule.
Yeah.
Tubule.
You point out a problem in this pathway crayons.
What?
There's problems with pathway.
You can never go back to the side.
Yeah, so this is a perfect representation as DM1 attaches the microtubules and blocks it because you can't add any more tubule.
And the stabilizer on the third part of the drug just keeps it together.
Yeah, so it's actually like a weakish bond, and it's broken down with lysosomes, so it breaks it apart and then releases the DM1 separate.
What happens to that third part? Just spread.
Which one is that?
What? Okay. You guys probably don't understand like what this graph is saying. It just looks like a bunch of mumbo jumbo, right? So dynamicity or dynamic instability or microtubule dynamicity is just the growing and shrinking of microtubules, and that is done by the polymerization of the tubule. So what this graph is saying is as microtubules move around less and less with suppression of dynamicity, the metabolite concentration goes up. So what is metabolite? Metabolite is just gonna walk over here. Metabolite is the products of metabolism, and a main process in metabolism is the breaking down of things in lysosomes. So as basically this is saying is as more DM1 is released through the breaking down of catacyla, more microtubules are suppressed.
Anybody have any questions on that?
Yep.
Because so there's two different lines there.
So these squares is just metabolite concentration, and circle lines is suppression of factors.
So basically, as there's more DM1, there's more suppression of adding tubulin, and then there's more metabolism.
Julia?
Why do you need like the part that inhibits like the microtubules and like another part that inhibits HER2? Why can't you only do one?
I mean, you can. The trastuzumab is just a way to get DM1 into the cell. They both inhibit cell proliferation. And basically, DM1 inhibits mitosis, and trastuzumab inhibits cell proliferation.
It's
It's just multiple ways to do the same thing, like multiple prongs. It's attacking it from multiple directions.
Sweet.
If trastuzumab binds to HER2,
Yeah.
how does and trastuzumab is attached to DM1?
Lysosome is breaking down inside the cell. It's internalized.
How does it get inside the cell?
Receptor-mediated endocytosis. So as trastuzumab attaches to the HER2 receptor, the cell just takes it into receptor-mediated endocytosis.
Any other questions?
Can you go back one slide? Oh, sorry, forward. And back. Yeah. We got it. Can you explain the second and third graph?
Which one? This one? Both? So this one, 4D5, this just shows that 4D5 is binding to HER2 instead of HER1 or some other receptors.
So why is it slightly binds to the HR1?
No,
Why
it
is
just shows that one of these binds to HER1. 4D5, which is trastuzumab, just binds to HER2.
Okay.
This one just shows, so all these dots represent different plates with cells in them. And they all started at 0, and they had time to grow.
8 days.
Yeah, and they had time to grow, and this is the amount of antibody given. So for different amounts of 45 was given to or put into the cells in the plates, and this just shows how much proliferation happened inside the plates. So with more 45, there's less proliferation because there's less cells. That makes sense. Any other questions, Amy?
In the first graph on this slide, what's the dot?
This.
Yeah.
It just shows like amount of, like the darker the dot is, the higher concentration of pairs of 4D5 and HER2 were found. Any other questions?